that'll help. So that reminds me to tell all the speakers, be sure you have the mic or people asking you questions, mic in hand, ready to record, because we are recording everything. We want this available for continuing education. So it's really important that you have a mic whenever you're going to contribute, be it a question. Of course, the speakers will be here. We have a lot of speakers that are remote. As a matter of fact, Ed Thomas with the Fertilizer Institute is not very remote. He's stuck in Dallas. So Ed's going to try to do it from the airport. He was supposed to have a recording. I don't think he does, but he has all the abilities back in Washington at the office. They have a full group, so he will, he can get it. He'll give the presentation, have a video, and that'll be available online later on. But we're hoping to have him here today. There may be a plane takeoff or so in the back, but we'll get it done. Dr. Karl Wyant, who is the director of director of agronomy at Nutrien. It's a leadership role in premium fertilizer product line and in advanced sustainability. Before Nutrien, Karl was the VP of Agri-Science at, how do you pronounce Heliag Agriculture in California and Arizona. Karl is a CCA and a certified professional agronomist and a licensed PCA in California and Arizona. Karl and his wife currently reside in Tempe, and they probably wish he was there today. It's a little warmer, I'm sure. Karl, thank you.
Thank you, Jim. Yeah, it was quite the shock when I walked out of the airport today. I did wish I was back in Tempe. Thanks for coming. I appreciate it. I wanted to share some thoughts with you, some things I learned along the way with biostimulants. I don't know if you ever had this experience trying to sell something, but management hands you a jug of something and they say, all right, go sell this. That was my introduction to biostimulants from the technical agronomy side, a big mystery box, right? And so we had to figure out the science along the way. And then I switched sides of the table. I went and worked at a biostimulant company and took some of that experience along the way and said, okay, how can we help identify the value proposition? What knowledge gaps can we start to fill in to help folks feel more comfortable with what we're selling? I work now for Nutrien, and not the retail side or the proprietary product side, but the fertilizer side. We're the largest mining and manufacturer of NPK in the globe, and so now a question that I constantly get asked and ask my team is, how do we innovate fertilizers? How do we take urea and take it to that next level? Potash 0060, 1034O, these basic ingredients we all know well and have been working with for a long time. And I think, and I feel strongly that biostimulants and biologicals are the way forward here. It's a way to innovate something that we already know. So my main, and probably the height of my artistic skill set is right here, where we have our current fertilizers, but then we add these new ingredients. We make them different, and we make them what I call fertilizer 2.0 or 3.0. Some of you might be 4.0 by now. I don't know. We don't need to have a contest about the version. But I think it's important. It's to give these ingredients a chance and say, okay, they're new. They're new to a lot of us. They're new to the industry. They're new to our customers and our growers. And how do we work through this confidently to make this a reality, to get more out of our fertilizer systems? So, we have these ingredients, plant biostimulants, plant biologicals, whatever your favorite slang term is, as long as it's a clean slang term, I hope. We have these things we can apply, and we get a result. Yield is what we're often most judged on. in terms of our programs and our product selection. And how we get there has been a little bit of a mystery. Is it through improving nutrient efficiency? Is it by improving stress tolerance? You know, I came up in the ag world in Arizona, California, lots of heat, lots of salts, and fairly low quality soils. And so we have a nice lab for that stress tolerance. And that was my introduction to this whole space. One of the challenges is you know, not just here's the product and how does it work and what effect does it cause. One of the real challenges we've had in the last 10 years is just the explosion of the space. Now, I put this chart on here, and I know I break all the rules of presentations by giving you a chart you really have to squint at to read it. But this is the reality of this biostimulant biological landscape. We have across the top and along the side there in the black bars, you know what they do. Are they living? Are they bio-based substances? Are they crop and soil health? And then underlined in each circle is, you know, what a broad category of what they might be: a microbial, biopesticide, inoculants. And so we have all this stuff to choose from. It's kind of like the snack table back there. You know, you got to choose something. I chose the little pink candies with the strawberries on it, super solid. But some of you might like pretzels. And I think our customers are in the same spot. What do we do here? Like, there's so much out there. How can we possibly evaluate any of this and really get a handle on what's going on? And I really feel for the person who had to create this chart. That's a lot of logos to track down. It's probably a graphic designer at some point. And I think one thing that we're also dealing with is what we're sort of used to in the marketplace. Our business as usual across a lot of the ag sector is this consolidated space. Crop inputs as fertilizer, that's a consolidated space. Crop protection, consolidated space. So we have this market for much of our seed, our fertilizers, our crop protection, all dominated by just a couple of companies. This new stuff, biostimulants, biologicals, highly fragmented. It's like the opposite end of the spectrum. I showed you this chart earlier. That's what I'm talking about, a market that's not consolidated. There's a lot of little companies out there that are all asking for your attention and ultimately for your dollar. And I think we're having a hard time kind of moving between these two worlds that we're all used to, this consolidated market of the inputs and now the new stuff, the biostimulants and biologicals, which were much more fragmented. So how do we figure that out? I'm not quite sure, but there has been some movement up towards that consolidation. Lots of purchases lately of these biostimulant companies, the Syngentas of the world, and there's a lot of other examples of this. But we're starting to get some consolidation, especially within the last 2 years. So before I kind of walk through how I like to evaluate these products, because remember, I've been on both sides of that, trying to figure out how to put it into a program and then trying to sell it to an ag retailer. I like to think about, what does the product do? What is the utility of the product? And I'll start with the living organisms, the biologicals, that's what I call them, where we talk about species identity. The species is what's doing the work, or the mix of the species. So we'll briefly go through that so it doesn't become a microbiology lesson. And then on the non-living side, what I call the biostimulants, it's usually the carbon chemistry that determines the function. How big is the molecule? What are the charges? What's the C to N ratio, a measure of quality? And then if it's a food source for microbes, what is the food you're actually eating? You know, it's like that table back there. Are you eating pretzels or a bunch of candy? I think I saw beef jerky back there. All carbon, but very different food sources, right? Same with these biostimulants. So we'll start with this inoculant cheat sheet, and I'll start with the fungi first. These are super common in a lot of our specialty markets where we're using different strains of fungi, different genre of fungi. I'll just start with that blue box up in your left. The yeast and protein mixes. These mixes are fungi that are used as a nutrient delivery source, and they're bringing amino acids, a little bit of nitrogen, phosphate, and potassium, along with some other micronutrients. So it's a great way to boost that localized nutrition and impact the plant growth. If you go over one box, still on the top row, to trichoderma, these are like a defensive move. The trichoderma are a beneficial fungus that helps protect the plants against other pathogens. So depending on what you're looking for, what you're trying to get done, that goal, here you have two different tools. We'll go down one box to Aspergillus. This is a fungi that helps produce enzymes that break down plant fibers like cellulose, things like that. So if you're trying to decompose, let's just say, your corn stover at the end of the season, that could be a good product to use, something with Aspergillus in it. And then finally, and probably the most famous, maybe the, I don't know, Justin Bieber of the fungi world, the mycorrhizae. I should probably update that celebrity reference here one of these days. Taylor Swift of the fungi world, thank you, appreciate that. I almost said Kim Kardashian, but that's like 20 years ago. So mycorrhizae, very common, lots of products out there. And here with the mycorrhizae, you're putting out very specific strains that are trying to develop that symbiosis between the plant and that specific fungus, and that fungus helps that tree or that crop get more nutrients, get more water. In return, the plant pays it in sugars derived from photosynthesis. So you see, you have, if you break down your label into what's on there, that name that's in italics, and you kind of like, well, what does this product do? Here's a quick cheat sheet for fungi. Great way to sort out some of these products that are coming your way. We'll swap over to the bacteria, and I'll start with the Bradyrhizobia up in the upper left. These are also the Taylor Swift, I think, of the bacteria inoculant world. These are often applied with soybeans, alfalfa, any of your legume crops. We've been using these forever and with lots of success, where we're adding those infixing bacteria with the crop, with the seed, and helping to derive more infixation through the season. So that's a lot of folks' introduction to these inoculants or these living bacteria. And then it gets a lot more complicated from there. You slide over one to that dark blue, the Azospirillum clavacea. These have become really a lot more famous lately. Maybe like Doja Cat, if we want to take a celebrity reference. Thank you for laughing at that. The Doja Cat of the bacteria. Free-living bacteria that help with infixation on non-legumes, so crops like corn. So we've heard a lot about these types of products lately. A lot of folks are still working out that biology, working out the application in their cropping system. But those are the identities of the bacteria that do that kind of work. Provide free nitrogen from the air and give it to crops like corn, which is super special. Slide down one, you have Pseudomonas. These are bacteria that are known to help with pathogen control and help make nutrients more available in the soil. And you can extend that to the Bacillus as well in the yellow box. So Pseudomonas and Bacillus, if you're looking at a lot of these bugs in a jug, they're often those two genre of bacteria, Bacillus and Pseudomonas. And a lot of it's because they can help free up nutrients that are contained in the organic matter of the soil. And there's also some evidence that they can actually help the plant and form some relationships with the roots to help the plant get more nutrients as well. So quick little cheat sheet. You don't have to memorize it. I promise, there won't be a test. But I think the challenge with the living products, the inoculants or the biologicals, depending on what you like to call them, is making sure that they're viable when you use them. And that's been incredibly challenging. I did some work where we actually looked at label rates. This is how alive the product is. This is how many living cells you can expect, per unit. And then you actually go plate it out, and that's not often the case. How long has that product been sitting in the warehouse? How long was it exposed to the brutal blizzard of San Antonio of 2024 that we just experienced today and yesterday? I come from the opposite end. I came up in desert agriculture. Like, can this stuff survive the summer? When it's 118 degrees Fahrenheit in the warehouse, where we store things. Just a spoiler alert: most of them can't. So that's been a real challenge. It's just, can we keep it alive? There are some new technologies coming in this space where they're actually able to put that bacteria or that fungi in a spore, put it in a spore, which is a super hard shell, so that bacteria stays kind of in a dormant and really tough phase until you apply it to the soil. So that's been exciting to see that spore forming technology come along, I'd say in the last 5 years, relative to just the free swimming bacteria that we're kind of used to. They're super sensitive to the environment you store them in. I mentioned that earlier. And often when you put them out in the field, if you take them out and put them into a cold spring soil in Iowa, are they going to survive? And that's been a challenge. If you want to take a look at some of these label claims, this is how alive the mix is. How many living cells you can expect. It's colony forming units per gram. You can actually, there's some labs that you can send the product to, and they'll actually count it out for you and say, hey, it's this, and you can get an idea of how alive is the product. Another piece we've had is folks want to take these living products and they want to put it with things that are already going on in the field, like the fertilizer, the crop protection, and we're having some challenges with just blending. You expose it to another chemistry, does it stay alive? And if you don't have these products alive, is it going to do the work that you want it? So lots of great questions to ask yourself, starting with what's the end result, and then working your way backwards into, well, how do I get it in the field and keep it alive? I'll switch over to the non-living products. I used to say the dead products, but I had a marketing person get mad at me because I was too negative. So non-living, this is the word she preferred. But we're going to focus on this bottom bullet point, the carbon chemistry. A lot of these biostimulants are carbon molecules, and you can predict what they do based on their chemistry. And so we'll just walk through a couple of examples: the size, the charge, C to N ratio, and the macromolecule diversity. I'll start first with that bottom, the food source, where you're applying a non-living food source, and this is derived from a whole number of ingredients. As a biostimulant, you're feeding the microbes that are already in the soil, on the leaf, in the roots, and you're using that food source to promote a better microbiome. So I'll show you this example, and this picture really lit this product category up. 2016 in Nature Communications, big deal, but they're showing a soil. If you look all the way to the left, that's a soil, not even a soil, it's a kaolinite and sand mixture. I don't even know what you call it. It's like something you steal from the greenhouse. And they have that, and what they did is they added glucose weekly for 15 months, let those microbes go to work, and when microbes are alive and happy, they are secreting these glues that help stick your soils together. And if you look at the soil on the far right, that's that same kaolinite sand mixture at the end of 15 months. This is the power of the living microbiome in the soil if you give it a regular meal, as they did. And so this paper really caused a lot of products to come up. Feed the microbes, wake up what you already have in the soil. Lots of ingredients, molasses, sugars, microalgae. And this is where, if you're in one of these discussions about food sources as a product, ask what's in the food. Is it a hamburger, or is it a big bowl of wood chips? Right? That's carbon, but they both taste very different. I didn't personally try that last one, but you get what I'm saying. The food matters. What you eat matters, and the same with your microbiome. So that's a good question to ask: is what kind of food is it, and what is that? I call it macromolecule diversity. What's the percentage of protein? What's the percentage of fats? What's the percentage of carbohydrates? And that's a good way to tease apart that question. Just kind of going down this list of the non-living products, there's fulvic acids. And these products are honey-colored. They're derived from overburden on coal mines. And they're much, much smaller than humic acids. They have that nice, pretty honey color. And what they really have that's cool is a high CEC, a sticking capacity, CEC of between 500 and 600 units. That really helps them hold nutrients. But they're small, and this gives them another advantage, is that they can take that nutrient that they're holding, say it's a nitrate, an iron, phosphate, hold it, and they're so small they can actually be brought into the plant. So it's kind of like a natural complex here. And interesting set of products. Some states don't recognize the existence of fulvic acids. California is one of them. They just say, hey, that's another color of humic acid. You know, don't try to pull the wool over our eyes. We grow almonds. But some states say, hey, fulvic acid, unique category, and here's, depending on where you live, here's some information on how they work. And there's also some evidence that they stimulate plant root growth, probably through that nutrient carrier pathway. Seaweeds and kelps have been around for a long time. There's evidence that, you know, several thousand years ago, there were folks up in the coasts of northern France gathering seaweed off the English Channel and putting it on their crops and seeing a nice result. And this is a macroalgae, so it's a little bit different than say a microalgae or some of these other, you know, cyanobacteria type products. Two kind of famous ones: the brown algae, the Ascophyllum, and there's a couple more that are commonly farmed or collected. And one thing to think about when you think of seaweeds and kelps, think about a cocktail. Maybe you'll be having one downstairs tonight, I hope. But they're a cocktail of extracts. If you were to try to say it's a single ingredient, kind of like when you look on a crop protection label, there's the ingredient, that's glyphosate, boom, we're done. These seaweeds and kelps are often mixtures of lots of different molecules. They're a little harder to really pinpoint what's in it. But we know they help with plant growth. We know they help with yield. I use these a lot when I was managing crops during the desert summer, because I knew they could help with stress. You know, I could foliar spray a seaweed, and I could push through some of the 122 Fahrenheit degree highs we have down. Don't take a vacation there in August, by the way. Right now is the time to be in the desert. But that's what we know. We know the plant response. It's been well studied, but it's got some challenges with the regulatory. You know, is it a plant biostimulant? Does it need to go be regulated through the EPA? I think these questions are still kind of up in the air. I hope Ed Thomas gets to chime in, because he could give you a good update about where seaweeds are at. So we know what they do. We kind of know the end story. We don't really know how they get there because they're so variable. Another common type of products, again, non-living, enzymes. These have become a popular set of products lately, and there's a whole bunch of them. I'll give you an example here. Here's a little bacteria, hangs out in the soil. It needs phosphorus. It needs phosphorus to help generate DNA. It needs phosphorus to help generate the currency the bacteria wants to use to grow, the ATP. And so the bacteria is going to secrete phosphatase. That phosphatase is an enzyme that'll break down insoluble phosphate, say something that's bound to calcium, same mineral as your teeth, the mineral apatite, very insoluble. This enzyme can help do that and liberate that phosphorus for the bacteria's use. So someone much smarter than me said, well, why don't we just put down the enzymes, cut out the middleman, the bacteria, and just put out the enzymes themselves and get the function we want. So that's been a fun category of matching up these enzymes, producing them at scale, and being able to get the reaction. So I'll list a couple of these enzymes for you. Protease, that helps with protein breakdown. Beta-glucosidase, that helps with cellulose breakdown, so say like crop residue. Amidase and urease, those are important in the end cycle. You might recognize that urease, that's the enzyme that's involved with breaking down that urea, turning it into ammonia. And then I mentioned phosphatase earlier, and there's sulfatase, that's for phosphorus and sulfur release. One thing I want to point out is that these enzymes have very specific functions. So depending on what you want to do in the soil and in the crop management, you've got to match it up with that active ingredient. What taste do you want to use? Is it glucosidase? Is it sulfatase? Some say tase. I know what you're saying. So that's one of the challenges with the categories, is matching up the right flavor to what you're trying to get done. So just keep that in mind. I'll continue with humic acids. And again, these are all coming from this overburden of coal. And humic acids are these big long chain molecules. I showed its close cousin fulvic acid. That's a small one. I had a carbon scientist, say that if fulvic acid is the size of a Volkswagen Bug, Herbie, a humic acid is a rail car 100 cars long. That's the size difference we're talking about in the soil. So very different sizes. Here's a rendering of what someone thinks an average humic acid looks like, because it's a little bit of a continuum. So, for you youngsters out there, great spring break tattoo, it's coming up. See you at Lake Havasu, right? So lots of stuff going on, big molecules, lots of functional groups, and this is what gives the humic acid such unique properties, different charges, different abilities to hold on to nutrients. And there's also some evidence that it can help with improving physical structure and also act as a plant hormone. So lots of different activities here. Not quite sure what category it does best. We're still working that out. But I will say one thing: if you were to try to eat a humic acid, you'd have a bad time, right? If you can have a hamburger or a big bowl of wood chips, this is the big bowl of wood chips. Lots and lots of carbon, not a lot of nitrogen, a poor quality food source. So watch out for that claim. I've done some work that supports that the food choice for the microbes. So what are the challenges with non-living products? One of the challenges is, I mentioned the cocktails. A lot of these non-living products are coming from these natural sources, and so they're not single active ingredients, one molecule like we're used to with crop protection. If you look at a fungicide label, there's your active ingredient. That's usually a synthetically derived molecule, and so you have really good predictive power. On the non-living products, they're usually cocktails of things. They're derived from a substance like a seaweed or a microalgae or a molasses. So you get a little bit of a mixture, and that can be challenging in terms of quality and its efficacy. There's also been issues with blending. I mentioned the fertilizers, 2.0, 3.0, 4.0. Folks are trying to put these non-living products in with their current fertilizers to make them even better. And there's been some challenges with blending. I'll show you an example here in a second. I think, and I kind of touched on this with the humic acids, if you look at some of the marketing, there's laundry lists of what these products do. And it's difficult to look at your favorite chemical rep, or ag retail rep, when they take you to lunch, right? And what does this product do best? And it's hard to get that force ranking of like, what does this product really do? And I think we're still figuring that out, the science, that sort of thing. I mentioned there's some need for logistics. You don't have to keep these alive usually. That's one benefit, but you do need to have them blend, and I'll show you that. And I think very strongly that because of the non-living factor, not having to keep this alive, like this can live in a warehouse, make it through a warehouse in Yuma, Arizona in the summer, it'll be all right. I think this path probably has, these types of ingredients have a good path within innovating fertilizers because of that. You don't have to worry about keeping it alive. So let me show you some examples. Here's a jar test that I did with a biostimulant product and UN32, right? Fertilizer 2.0. And the picture on the left side, that's when I made the mixture. Looks good, right? I feel very happy about sending it out into the field. Nice color. I got a little more margin on my blend. I got some more benefits to the UN32 because I did some Mr. Wizard chemistry there in the parking lot. But if I waited 48 hours, the mixture separated. And this is a challenge across these biostimulants, is just, does it stay in solution? And there's lots of products out there with this example that, does it stay in solution? Are you going to get that call at 6 in the morning on a Sunday, from your customer who's grumpy because it did what's on the right there? And that's always a challenge. So before you start making new mixtures with new products, those jar tests can really help you out. And I'd also say that these blending, we have some rules for fertilizer blending to make sure that things are safe. Don't mix bleach and ammonia products, right? That's not good. Even if you're just cleaning a tank with bleach and then you come back with an ammonia fertilizer later, you're going to have a bad time. So we've got all these safety rules. We've got these full compatibility charts, dry and liquid, for mixing fertilizers. But what we don't have is the safety for these new ingredients. And I think it's important to say that we've got to keep that at the forefront so that not only are we making blends that go together and stay together, but they're not causing any undue harm to customers, because there's just so much to choose from. I showed you that constellation of logos. How many of them have been tested with different fertilizer combinations for safety? I would say very few. So keep that in mind when you're making these, when you're trying to innovate fertilizer. We've got to keep safety in mind. And just showing some more examples. Here's another mixture I made. I made it, I didn't sell it. This is my one of my training jar tests that I have new agronomists do. They mix here in the middle, that's can 17 and 1034O. And so it just makes toothpaste. And I say, hey, if you send that out in the field, you're going to the farm on Memorial Day at 6 a.m. to unclog a tank or a bunch of nozzles. That's your bad. Do the jar test first, though. So I mentioned those compatibility charts earlier. Here's that example of if you look at this tiny print, if you're way in the back, that's listing all these different fertilizers. Green means they can mix with other fertilizers that are on the same color, and if they're red and yellow, it's not going to happen. But the new ingredients are not on this list, and I think that's a critical piece of information that we need to all remember, is the safety part. So I know this is small in the back, but this is just some of those safety measures. And if you look way at the bottom, there's one called mixing acids with humic substances. And the safety call out is carbon dioxide gas generation, and that can reduce or displace oxygen in the air and cause respiratory issues. I know it's hard to see. But this is one of those pieces. This is one of the first mentions I've ever seen of a biostimulant or biological for a safety concern. And here it is listed. So I think a great place to start is the material safety data sheet. Supplier advice can really help you with that safety piece, and the blending part, spec sheets, and my tried and true jar tests. I have a lot of jar tests. My wife loves it. They're just all over the house, because I need to know what's going to happen 5 years after I make it. Hopefully I label it. Then it's a real, if I don't, it's a real mystery. But that jar test, trying to figure out, predict, is this safe? Because I don't have much information to go on, either way. I saw some of you folks taking pictures. If I'm sitting at the front here, if you want to drop your card off, I'd be happy to send you this liquid fertilizer safety article I wrote. I could just send you the PDF, and it has that compatibility chart in there and that safety chart as well. So happy to share it with you. I'm not going to turn over your email address to like some spam service or something. Just want to kind of raise that conversation about safety more and more. So I'm sitting right over here if you want to drop your card off. Moving forward, consultants and advisors like the ones in this room, we're the ones that are going to have to tease all this information out and help our growers wade through this avalanche of products and claims and modes of action. And I think that's an important challenge to take on, and we've got to do it in the right way. Nuanced functionality, crowded marketplace. There's just so much to choose from. I always say, watch out for wild claims. Nothing gets a big eye roll from the agronomy side of the technical agronomy side of our business than just some of these wild claims that are made. And I won't repeat them up here. I have a really catty group chat between agronomists where we take photos of them and send them to each other. I always say, start with the end goal in mind. What function do you want to provide within your blend? How do you want to supercharge your fertilizer, and then start making your choice of ingredients? I mentioned safety and compatibility, super important. And I think regulatory changes, and hopefully Ed has a chance to leave the Dallas airport and talk about the regulatory piece and some of their work at TFI. But I think we're starting to get some closure on this wild west product space, and maybe some more rules. One piece I wanted to talk to on the performance is when you're looking at these biostimulant biological data, this is what you often see. We have the check field in blue, and then our product. And I just made this data up, product A. And here we're saying, in this example, we got an 8% yield bump and an 8-to-1 ROI. Or you can take that to net yield, net dollars per acre, or whatever you want to do. But this is a very favorable result. But as consultants, I think it's important to say, hey, what about the statistics, your error bars? What can you tell me with that? And that's where a lot of these marketing claims start to get a little shaky. Here I'm showing some non-significant statistical data here. And I think that's important to talk about because that's your customer's experience. Is not only the mean, the lift on the yield or whatever you're using to measure success, but it's that variation in the high and the low of the performance. And that's what your customer is really, I think, really looking for some advice on. It's like, how do I tighten up this experience and make this work for me? So watch out for that. So I just have 6 or 7 questions. I said 6. You can tell I went to Arizona State. Can't count. 7 questions. I say this is a great place to start with that person who's taking you to lunch to pitch you their product. What does your trial data look like? And they should have a good investment in trial data. And we all have different levels of satisfaction for what good looks like. But I think the days of just bringing one trial to the party and say, hey, you want to buy this? I think that's over. We got a lot more capital coming in to test. Do you have a clear story on your mode of action? How does your product work? How do you get from an application to the yield result? What's in between? That's an important thing to know, and they should have that story. Ask them to give you a probability of providing a positive return on investment. They should have enough trial data, and say on corn, that, hey, we have a 73% win rate that will on trials, and we can say on average that 73% chance of giving you this many bushels an acre over the grower standard. Something along those lines. They ask about patents and IP. Are they just a dime a dozen company, or are they really making a new novel product? What does their regulatory future look like? Some of these companies are dancing on that thin line between staying free of EPA regulation. Some are jumping in the pool. So just watch out what their regulatory future looks like so the product doesn't get pulled. And this goes back to the trial data and the mode of action. They should give you a good enough story where you see it, the fit. In your library of products, you see where that book goes, that book on the shelf, where the space is. And I think that's an important conversation to have. And then finally, and we've seen this lately, venture capital. How long are they going to be around? Are they well-funded, or are the doors shuttering in 6 months? We've seen some folks get burned pretty bad lately as the global markets have changed, and these VC sources have, I think they realize that ag's a different business than a forever going up J curve on product sales. So some just important questions to ask. That's my last slide. I'll take any questions. And just a reminder, if you want that article about the safety, feel free to drop your card off at the table here in the front, and I'd be more than happy to send it to you. Thank you.
Thanks, Karl. Are there any questions, comments? And we're going to get the, I think Karl's last slide there really summed up, or his last information slide really summed up where we're at. We have a lot to do, learn how to use these products. Anyone have any experience? Steve? Steve's always got something to say.
Thank you. Just a question on the slide that you showed about that paper from Nature, where
Yeah.
you have kaolinite already sterilized with sand, and then you added molasses or whatever you added there, and then you ended up having highly structured or good structured soil.
Yeah.
Is that a function of the additive that you added? Because that's we expect microorganisms to be still there to do the work. But if you sterilize the kaolinite and a mixture, where would the organisms come from? Is that from the additives or amendments?
It's thought that the structure change is coming from the substances being released by the microbes that either come in because the mixture is sterile, but they can't the glucose additions, the air around it has microbes, so they're feeding something in the soil. And if this is a good study where it's the microbes themselves that are secreting the glue, the polysaccharides that are sticking the particles together and giving that change that you see.
Okay, thank you. I just want to make sure that microbes have a lot to do with why you have that highly structured.
Yes, it's the microbes that are driving soil structure, fungi and bacteria.
By the way, my name is Leticia Sonon. I'm with Uber Agro Solutions.
Oh, thank you. Appreciate that. Great question.
So outside of humics, which of the categories have you had the most success with in adaptation and adoption from the marketplace?
Another Arizona guy, right? I think I applied for a job with you like 10 years ago. Small world. I've really liked the humics, but challenge with the humics is sometimes in those big liquid tanks, they'll separate out. So I really got into Fulvics for a while, and the Fulvic-like products that are sold in California because there's no label for them. So I've really enjoyed the Fulvics. I also worked for a company that we did these microbial foods, and I had a lot of fun with that, and we saw some cool success testing those products. And I think the thing I've had the most success with is sales team that has come to me and said, how do I use a seaweed versus a food source versus this? And just trying to figure all that out. And I think maybe it's not the product itself, the active ingredient itself, where I've had the most success is finding where the slot is. Like it's like crop protection. We have very specific slots to fill with our products. I think these biostimulants, biologicals are going to go the same way. We're just finding this right time in the right place. That'll give you a much better experience. Thanks, Steve.
Yeah, no, sure. We've tested a lot of them, of course, and yeah, finding the right slot is the key. Figuring out how to test it so it can perform is huge.
Yeah, good question.
Remember to say where you're from and what you're working on.
I'm Kelly Chapman. I work for Global Agricultural Development Corporation out of North Carolina. And we assist companies with the process. And so often we run into the thought process that you can skip steps or really speed up the development process. And I guess I would just ask you, what do you say to younger startup companies that talk to you about that process? Or how would you convince them that you can't skip some of these steps or you wind up with a problem?
Great question. Which step can't you skip? All these are important, but I think it comes down to culture. And this orange box down at the bottom, your funding source needs to have the patience to not see a J curve, sales go through the roof in year 2. That this is ag. It's based on seasons. It's based on relationships that are hard won and earned through reputation and reliability. And that this is a long-term game we're playing, and that the board or whomever is writing the check, I think, needs to maybe know about the culture of agriculture. I don't care where you go in the world, it's all kind of slow relative to venture capital, and I think that's okay. This is a food source we're talking about. We don't want to gamble. We want to make sure our relationships last and our reputation stays true.
Just as a follow-up to that, then there's a lot of discussion and debate about like greenhouse trials versus not, small plots versus strip trials, etc. Nutrien's been very successful. So what do you think, or what's your opinion on what's required for development?
Good question. I think greenhouses are a great place to start, But unless your market's the greenhouse, you've got to move outside. And that's where it gets a little trickier, where the people like me who are evaluating the statistical nerd stuff, we want small plot replicated trials across the big geography using similar methods. So that's one market, to convince that first set of gatekeepers that, hey, this product has legs. But then oftentimes when you get to the ag retail or the grower level, they want to see, oh, let's just split a field in half and see what happens. And so I think you've got to commit to different trial types per different audience. Who are you telling a story to? Are you telling it to the agronomist, or are you talking to a customer who doesn't have time to go babysit all these small plots? Good question.
So I'm reminded, along the strip plot side, I'm reminded of a talk that I listened to Marcus Jones talk about. Do you know Marcus? Have you worked with Marcus? He came out of Iowa and he was at Winfield doing a lot of the answer plots. He said they took a homogeneous acre in the middle of one of the I states, wherever it was, and they thought it was perfectly uniform soils. They planted corn on the whole block, and they harvested it one row 30 foot at a time. And across that block, they had somewhere between 150 bushel and 210 on a block that should have been perfectly uniform. And that was also, then where Marcus started talking about every trial has at least 2 gradients that we don't know about and so forth. But so when we talk about strip trials, and I've done my share of them, and I hate them. For an extra $10, your stuff will look really good and the other guys will suck. But it's hard.
Yeah. But how many customers want to do the strip trial, right? Well, let's try out this year. Let's do a strip trial. Yeah. Every testing protocol has its pros and cons. How do you get around it? I think that's a moving target based on who your audience is. Good story. Anything else? Awesome. Thanks for having me.
Thank you, Karl. I'm going to remind you that in the front here, GAIAC is having a membership drive. We're trying to spread our awareness. We have a lot of candidates. We have a WhatsApp chat room with a global community. So we encourage you to sign up, join. There's a QR code up in the front. Take you right to the sign-up sheet. And I'm also going to have there'll be some other things that'll link us to, or will link you to some articles on regulatory processes in the EU. So, and I'll mention those again. So while you're taking a break, come on up and join us at GAIAC. Thanks.